My name is David Hess, and I'm a scientist here in the beautiful Krembil Center for Stem Cell Biology. I'm a member of the Vascular Biology Research Group, and as such, I work with stem cells to look for alternative therapies for vascular disease and to treat diabetes. These are some of the students that are working in the lab. They're taking a look at a specialized type of stem cell called endothelial stem cells that are involved in vascular repair or the formation of new blood vessels. And in the lab, we are trying to do that to treat a disease called critical limb ischemia. These are patients that have such poor blood flow in their limbs that they are at risk of amputation if nothing is done. And cellular therapies could be the wave of the future using endothelial stem cells or endothelial progenitor cells to help create new blood vessels to save their ischemic limbs. The hard part about working with stem cells, and we work with what's called adult stem cells that come from human umbilical cord blood or human bone marrow samples, is that they are very rare. And there are multiple different types, all with different functions and different jobs within your body. To find this needle in a haystack, I call it, this 1 in 1 million cell, we have specialized technologies. And the first step in finding the needle in a haystack is to use this very cool machine called a RoboSep. And this will take our 200 to 300 million umbilical cord blood cells and pick out only the primitive cells, let's say 10 million cells, and within that population, a couple 100,000 will be stem cells. So the next step in the process is to use a very specialized piece of machinery called a fluorescence activated cell sorter that can sort through these 10 million cells at tens of thousands of cells per second and put the stem cells in one tube that we will use for our experiments and put the non-stem cells in another tube that we use for comparison. Let's go and take a look at that. Right next to our tissue culture stem cell facility is the Robarts flow cytometry facility. And in this room, we do the final steps in preparing our samples for our experiments, in isolating that needle in a haystack. This is Kristen Chadwick, and she runs the flow cytometer. And what this machine in effect does is it shines a laser on the cells, decides whether the cell is a stem cell or not, and will sort all of the stem cells into one tube and all of the non-stem cells into another tube. The amazing thing about this machine is that it can do it at 10,000 cells per second. So essentially, we end up with a purified population of stem cells for our experiments, looking at treatments for diabetes and treatments for vascular disease. So that's what we do here at the Krembil Center for Stem Cell Biology at the Robarts Research Institute. Thank you for coming on this tour, and I hope you have a great day.